Good morning, everyone. Thank you for joining my presentation, which will be mainly focused on using next-generation technology and big data for research in the forestry sector. This presentation is only a snapshot of all the research conducted by the TPCP CPHB group. I would like to start with a definition of the Fourth Industrial Revolution. According to the World Economic Forum, the Fourth Industrial Revolution represents a fundamental change in the way we live, work, and relate to one another. It is a new chapter in human development enabled by extraordinary technology advances. What we need to keep in mind is that the Fourth Industrial Revolution does not only transform industry and society, but that it is also transforming research. The World Economic Forum therefore wrote in its policy document that scientists should harness the Fourth Industrial Revolution as a positive force for, on the one hand, managing and conserving life on land, while on the other hand, mitigating the risks that advanced developments in science and technology might create for life on Earth. Scientists that are part of the Fourth Industrial Revolution are therefore laying first of all a foundation by generating data using new technologies and platforms. Building upon this by developing new methods to access relevant and useful information from the massive datasets created by next-generation technologies, and finally and most importantly, this information needs to be applied for sustaining and improving life on Earth. The TPCP CPHB has a long history of making use of next-generation technologies to generate big data sets. These techniques include genomics as well as transcriptomics. During the last few years, we have also made considerable progress in getting a metabolomics platform established. Currently, we are digitizing our extensive disease incidence records in order to build big databases that can be used as a platform for modeling and diagnostics. All of these established and new platforms are assisting to develop new ways in which we can control pests and diseases in the South African forestry sector and keeping trees healthy. The TPCP CPHB has a long history of sequencing genomes. Brenda and her team have been sequencing the genomes of more than 50 species of fungal pathogens which cause disease on trees. We have also sequenced the genomes of all the major insect pests that cause losses in South African plantations. I will now show a few examples of how the genetic information of these pests and pathogens are used by our team to generate novel methods to solve pest and disease problems for our stakeholders. The first fungal genome that the TPCP CPHB published in 2012 was that of Fusarium cycinatum, and this picture commemorates the collective effort that was put in. Since then, at least another 130 genomes from different strains of this fungus have been sequenced. Fusarium cycinatum is a fungal pathogen in pine nurseries where it causes root and collar rot of seedlings. It also causes a severe disease known as pitch canker in the field. Having genomic information of a large number of distinct fungal strains is a powerful tool that can be used to get relevant and useful information from large data sets by inference. One example is finding fungal hormones that control certain developmental processes. For example, fungal pheromones that are used to identify a mating partner can stop fungal growth. Using the genomic information of Fusarium cycinatum, the alpha pheromone of this fungus was identified. Applying the alpha pheromone in different concentrations to pine seedlings during inoculation with Fusarium cycinatum reduced their infection dramatically. As can be seen here in the graph, a concentration higher than 5 millimolar almost inhibits the fungus entirely. By doing some targeted experiments, we realized that Fusarium cycinatum has developed resistance to some fungicides. At the moment, using the genetic resources available for this fungus, we are elucidating the mechanism of how the fungus is circumventing the toxicity of these fungicides. Information like this is really helpful in adjusting control strategies. Using the genomic information available for Fusarium cycinatum, fungal transcription inside resistant and susceptible hosts was analyzed. This showed us very clearly that a resistant tree can attack the fungus as well. One way in which the tree attacks the fungus is by inhibiting fungal lipid biosynthesis. Ergosterol is a fungal lipid that is important in the fungal cell membrane. Eric Visser and Sanushka Naidu showed very convincingly that in resistant Pinus techumanii, multiple steps in the fungal ergosterol biosynthesis pathway are inhibited. Ergosterol is specific to fungi, and therefore it is a good target for gene silencing. Gene silencing works by applying a double-stranded RNA of a fungal gene sequence to the plant. The plant recognizes this foreign RNA and mounts a very complex immune response. Once the plant's immune system is primed, it can recognize fungal RNA at any time and attacks it whenever it occurs on the plant. This could become one of the most promising and sustainable control strategies for the future, not only for Fusarium cycinatum, but also for a lot of other pathogens in forestry. I have now highlighted one example, Fusarium cycinatum. However, more model systems are under development. These include Ceratocystis causing wattle and eucalyptus wilt. For these species, we have already more than 200 genomes. We are also focusing very strongly on Teratosferia destructans, the causal agent of eucalyptus shoot and leaf blight. One emerging pathogen, which is going to become really important in future, is Austropuccinia psidiae, the causal agent of myrtle rust. To be on the forefront of developments concerning this fungus, we have already sequenced its genome, which was the biggest fungal genome that we have sequenced up to this date. As I mentioned earlier, we have sequenced 6 genomes from the most important forestry pests in South Africa. These genomes can now be used for a lot of practical applications. To illustrate this, I will show the progress that has already been made by our team on the Cyrex wood wasp. The Cyrex wood wasp kills pine trees by laying eggs together with a toxin into the stems of adult trees. Affected trees subsequently decline and die. Brett showed the biocontrol strategy used against this pest. However, it is important to have more than one strategy in place for controlling an insect. A good augmentative strategy for controlling Cyrex could be mass trapping. However, this is a difficult undertaking as little is known about how to trap these insects. The first thing to consider in a trap is the lure. An insect lure can include cues from the environment such as the host tree. Good lures often also contain insect pheromones, which are produced to attract a mating partner. However, as this figure illustrates, the possibilities of what can potentially be used as a lure are endless, and identifying a combination that works well is complex. To help with finding a lure, a small team is working extremely hard on studying the chemosensation of the wood wasp. Chemosensation is driven by a group of proteins known as chemoreceptors. Our group have identified all the chemoreceptors in the Cyrex wood wasp, and by looking at different body parts of the wasp, a heat map was drawn up looking at the relative expression of these chemoreceptors at different life stages and under different conditions. This already reduced the complexity of the data, as now we can focus specifically on, for example, this region of the heat map if we want to identify which smells attract or repels male adults. However, if we compare the expression patterns between female and male antennae, our analysis actually reduced the complexity by 95% because there are only two clear candidates here, where females have very low expression and males have very high. These are very exciting results, which leaves us with promising candidates for future studies. Apart from the lure going into a trap, the external design of a trap should also be considered. In order to determine which color should be used for a trap, the genes for color vision were extracted from the Cyrex genome. Based on this genomic information, as well as some other electrophysiological measurements, Cyrex can only see green. Compared to a human who can see red, green and blue, this is not a lot. But Cyrex can also see in the UV range, and this is very important, as you will see just now. Since Cyrex can see green, field experiments were conducted to determine if the insects will fly to a green trap. However, as you can see from this data, the green traps elicited the same response as the other colors. Therefore, we had to look into the UV range. To do this, traps were placed at specific angles to the sun. And this actually really gave us good results. Traps that catch the morning sun at 36 degrees northeast were much more attractive than traps placed at other aspects. Therefore, the insects are morning active and respond to reflections in the UV range. Future traps should therefore be placed so that they catch the mid-morning sun. This research project very nicely illustrates that by looking at the genomic information of Cyrex, we were able to ask the right scientific questions and design experiments that would give us relevant information. I have now highlighted one example, Cyrex noctilio. However, more model systems are under development and are providing insights into possible new control strategies. These include Coniferous species 2 causing defoliation of eucalyptus, its egg parasitoid Anaphis nitans, and the eucalyptus gall wasp Leptocybie invasa. Next, I would like to focus on next generation pest and disease control strategies that we are developing at the TPCP CPHB. Gene drives consist of genetic elements that are effectively transmitted among sexually reproducing individuals, even if they reduce an organism's fitness. Gene drives can be used to introduce a number of different gene traits to directly control the numbers of a pest species. As such, this could be an extremely effective technique, but implementation with previously available techniques for making transgenic insects and fungi was not straightforward. A new technique known as CRISPR-Cas9 gene editing has changed that. This technique can now be used to introduce mutations into a parent generation, allowing rapid spread of the altered gene in a population. We have now a number of researchers working in the TPCP CPHB developing these techniques for insects as well as for fungi. Gudrun has also won a very prestigious research fellowship from the Royal Society for developing ideas for applying gene drives in the South African forestry sector. Another important area of next generation pest and disease control in which we are currently starting to develop capacity is data science. In collaboration with data scientists from the University of Pretoria and Ilaria Germes-Huysen from the ICFR, the TPCP has started a number of different data science projects. These projects are currently still focused on building integrated databases and curating existing data to become useful for practical applications. We currently have a group of data science students modeling the population dynamics of Coniferous in the field. We have also invested in equipment and capacity to do multispectral imaging for early diagnostics of disease outbreaks. These techniques will also become very useful when screening for disease resistance of industry planting stock. We have started to capture all our data from the last 25 years to create a diagnostic clinic database. More than 50 students and staff members were involved in capturing more than 15,000 paper records electronically. We are doing this to develop a pre-diagnosis tool for foresters to use in the field and to process samples in the diagnostic clinic. Through generous government and industry funding, we are currently building state-of-the-art plant growth facilities for industry screening projects of pine and eucalyptus to determine their resistance to various diseases such as pitch canker, Teratospherias stem and leaf blight, and myrtle rust. This facility will be located on the experimental farm of the university. Varieties developed today will be standing in the field 50 to 100 years from now. If we can predict their tolerance to pests and diseases under future climatic conditions, this knowledge will be very valuable for our stakeholders. The facility that we are building will therefore consist of 3 independent compartments, which can be operated independently to allow for different climate regimes. This will allow us to do research on the effect of climate change on disease susceptibility or tolerance of current and future industry planting stock. This is the construction site of the greenhouse facility, and construction is going ahead even under phase 4 lockdown. Finally, in order to make it easier for our stakeholders, we are now in the second phase of development of a mobile app. This app will make sample submissions and pest and disease monitoring easier in the future. The first phase of this app was developed by third-year computer science students from the university. We are now using a professional developer to iron out some problems before releasing it to our stakeholders. In this presentation, I started off with the Fourth Industrial Revolution and its pivotal principles and expectations from research. I hope I have succeeded in showing that the TPCP CPHB group generates data using new technologies and platforms, and how all the different disciplines work together to access relevant and useful information from these massive data sets, which can now be applied for keeping trees healthy. We are very lucky that our program is built on the solid grounding of the forestry industry of South Africa, which enables us to do cutting-edge research. In this presentation, I could only touch on a few research projects, but there is much more and equally exciting research going on which I couldn't mention due to time constraints. I hope that next year we will be able to meet again in person, so that there will be more time and opportunity for each of our students and staff to talk about their own research and to let the enthusiasm of their research and underlying goals shine through. Thank you for your attention.